Hi, hello, and welcome again. Microbehunter here. I'm Oliver, and welcome to another microscopy podcast. Just recently I started to start the series. I started to start the series. It's a little bit of a word repetition here. I started this podcast series with the intention of answering your questions. And for the next half an hour or so, I would like to indeed answer again a couple of questions that are relating to microscopy or microbiology, microbes in general. And today I'm very happy to present to you a few questions that were sent to me by a 12-year-old fan of my YouTube channel. And he came up with, six, seven, 8 questions. And these questions are actually quite interesting, and I would like to share them with you, and I would like to go a little bit more in depth here and answer them. And they go all across the board, so to say, about microorganisms, also a little bit about the environments in which certain microorganisms can be found, and so on. And I thought I'm just going to dive into the topic a little bit and share them with you because we can all learn from this. I'm going to start off with the first question right now. It sounds interesting here. Can microorganisms talk or communicate with each other? It might surprise you. The answer is yes. Microorganisms can indeed communicate with each other, and they do that by sending out chemical signals. They are also able to detect their own density, basically how many of them are currently present. And this technique or method is called quorum sensing, written with a Q, quorum sensing. And what they do is the cells release certain chemicals, and when there are many of these cells around in a certain environment, then the chemicals increase in concentration, and this is how they're able to detect how many of them are actually around. Why is this relevant? Well, if they have reached a certain density, then they can actually do certain things, cooperate and do certain things that they would have not done as individual cells. So, for example, they can aggregate together and they can switch their metabolism around to produce different chemicals, maybe for the breakdown of food and so on. So they will actually change around their biochemistry depending on the number of cells that are currently present. So yes, the short answer is yes, there is indeed communication going on. Another quite well-known example, and these are not bacteria, but these are eukaryotic cells. That would be the so-called the slime molds, also myxomycetes, they're called. And these cells, they also release chemical substances, CAMP, cyclic adenosine monophosphate. It's also, again, a pretty fancy word. And this CAMP actually is used by these cells to coordinate. And if there are many of them together, then they will start to not only to fuse together, but they will actually also start to make so-called little fruiting bodies and produce spores. So it's kind of interesting to see that these slime molds essentially are kind of an interesting transition zone between single-cell and multicellular organisms, and they communicate with each other using this CAMP molecule. Yeah, so this is actually something quite fascinating. But we do not even have to go into the microorganisms. Let's just look into our own body. The cells of our immune system, for example, they also communicate with each other over chemicals. Yeah, so this is actually a fundamental principle that can be found quite widely in biology. So I think that this question is quite interesting and relevant here. But I'd like to now move on to the next question. And this is also something that I have talked about quite a few times already. Here's the question: are there more microorganisms on the human body than human cells? So are there more microorganisms on our body and in our body than we have body cells? And of course, yes. The answer is a clear yes. The estimates, they kind of range. I've read some sources that said that we have approximately 10 times as many bacteria growing on our skin and in our digestive system than we have body cells. I've then again read that this is probably a little bit overestimated, and that probably the numbers are approximately equal. Now, the more interesting question is how is it possible? How can there be so many cells in our body that are essentially not our own body cells? And the answer is that bacteria, for example, they have on average a diameter which is 10 times smaller than a body cell. So let's just say that one of our body cells has a diameter of, let's say, around 10 micrometers. A bacterial cell only has a diameter of 1 micrometer. But this actually means that the volume of a bacterium is 1,000 times smaller. So length times width times height, so 10 times 10 times 10, 1/10 times 1/10 times 1/10 is 1/1000th. So this means that bacteria actually are 1,000. One bacterial cell is 1,000 times smaller in volume and also in mass compared to a body cell. So this can actually show that it's quite well possible that there are way more bacteria growing in our intestine, for example, than we have body cells. As a matter of fact, bacteria are extremely important on our skin to protect us also from other harmful bacteria. So those bacteria that are present on our body is actually beneficial in that sense. And also in our digestive system, these bacteria, they help digest the food. So this is actually called the biome, the body's biome. And in recent years, there have been quite a lot of studies done concerning the body's biome. And I personally, from a philosophical standpoint, consider it kind of fascinating that we're actually made up of more cells that are actually not our own body cells. But actually now you could consider them being part of our body as well. Yeah, so it's interesting, really. So this means that we have a lot of also foreign DNA or non-human DNA on our body and in our body, which basically is also important because those bacteria that live in our intestine, for example, they are able to digest food and they bring along enzymes and are able to help in digestion, digesting food that we probably normally would not be able to digest. So you see that there is a very close cooperation, even symbiosis between our human body and the bacteria that live on us and in our body. And if you've just already put a little bit of saliva under the microscope, you're going to see how many bacteria there are. It's really incredible, right? So the answer is clearly yes. There are more microorganisms, specifically bacteria, on the body than we have body cells. So the next one is and one that talks about Stentors. Where do Stentors live? I could not find any info about it in books. This is what the question was. What's a Stentor? Stentor is a microorganism made of one single cell. It is a ciliate, and it basically grows in water, in fresh water, on the surface of rocks and also plants. So in that sense, it is able to move around. Yeah, so it's able. Some of them is able to break off and swim around until they reattach. But what they do is they filter the water for bacteria and other debris, and they have cilia. And they will therefore pick up any solid materials that are sufficiently small, and they will absorb them and will feed on them. So if you want to find Stentors or any other microorganisms, do not put the water directly under the microscope, but rather try to find some biofilm. Basically, this slimy layer that you can find on rocks in a pond or on plants, on decaying material. And if you're a little bit lucky, you're able to find them there. Now, the species Stentor was described in the year 1815, and there are currently around 20 different species that are described. And it's basically those Stentors are generally they're also known as trumpet animalcules. It was the traditional term. Obviously, they're not animals. Being ciliates, they belong to the protozoans. Essentially they're filter feeders. For me, always a little bit surprising that cells can look so extremely different from each other. But then when you zoom in and when you actually look at the details, then you're going to see that there is a big similarity among different creatures in any case. So let's move on here. The next question is a little bit more serious here. It's also a very important one. Maybe I'm going to spend slightly more time on that. Which microorganisms can cause death and disease? Okay, so this is actually a very long question, and to give you an overview, these are so-called pathogens, and pathogens are basically microorganisms or viruses. Viruses specifically do not count to the microorganisms because they're not alive, but microorganisms that or viruses that essentially can cause diseases, and you distinguish them between bacteria. These are basically here those microorganisms, single-celled microorganisms without a nucleus. You have protozoans. Yeah, for example, Stentor is a protozoan, but of course it's not a pathogen. And these are eukaryotes, so they have a nucleus. We also have fungi as possible pathogens as well. Yeah, we've got then of course also the viruses, which officially do not count as organisms. Yes, and those pathogens are able to grow on our body and in our body. So specifically when we have a wound, for example, and then the physical barrier, protective barrier is not there anymore, and then those pathogens can enter the body, and when they will start to reproduce, and they're able to produce certain toxins, which are chemicals, poisons, which can then harm the body. I mean, that's only one of the ways how basically they can be dangerous, and essentially different pathogens are quite strongly linked to the disease that they cause. So for example, malaria, the disease malaria is caused by a protozoan called Plasmodium. Yeah, and important is that there are certain diseases always caused by a specific pathogen, right? So that's this link is very important. So if you go back in time, for example, in the Black Plague in the Middle Ages, was caused by a bacterium called Yersinia pestis. Cholera is caused by a bacterium, Vibrio cholerae, and so on. So there are many bacteria around, and these bacteria can be treated with antibiotics. And it's a little bit more difficult when we talk about yeasts and, for example, which are fungi. So there are also yeast infections. And because those yeasts or fungi, they have a nucleus similar to us human beings. They're eukaryotes, and for this reason, the biochemistry is much more similar to the cells of our body, and therefore it's more difficult as well to find antibiotics that work against protozoans, which also have a nucleus, and fungi, for example, because their cell metabolism is much more similar to this of a human being. To give you a more specific example, the first antibiotic, which was penicillin, is able to attack the cell wall of bacteria. And we humans don't have a cell wall, and therefore it of course works. But we therefore have to be always to make sure that those antibiotics that are being made, that they do not have any side effects, and that they only attack those metabolic pathways in the pathogens that we do not have. So this is quite an interesting thing here. I did not talk about viruses yet. Viruses, for example, you cannot use antibiotics for viruses because viruses do not have their own metabolism. So what viruses have to do is they have to invade a body cell, and then they reproduce inside the body cell by using the body cell's own system of copying. So the DNA is copied, or the DNA of the virus is copied, and so on. And this is done by the metabolism of the cell. So in that sense, antibiotics that work against bacteria cannot work against viruses, because the viruses essentially simply use the metabolism of the body cell. Yeah. So this is if it says here which microorganisms cause death and diseases, is a question. Well, the list is quite long, but I have to say at the end that of course the majority of microorganisms do not pose any danger here. So what is the most extreme environment they have been found in? Well, it's a superlative most extreme environment. According to my research, they have, especially bacteria. Some bacteria can be extremely resilient, can withstand water in volcanic areas, which is nearly boiling, and also very acidic. Yeah, so this is a very extreme environment, and also bacteria have been found, also believe it or not, in nuclear reactors in places of very high radiation. So these bacteria have then obviously must have a very sophisticated DNA repair mechanism, because high radiation actually destroys the DNA of cells. For this reason, radiation has already been used and is used to disinfect, for example, surgical tubing, surgical plastic equipment, and so on, usually disposables. And what you do is if you, for example, have a plastic syringe and you want to get it sterile, it's impossible to make it completely sterile because bacteria are all over the place. And when you package it, then you're of course also going to package some bacteria, inadvertently. So how can you actually sterilize them? So what they do is they send this equipment through a reactor with high radiation. And this will basically like X-ray, the equipment after it's been packaged. So the whole thing is already packaged in a box. It's going to be sent through a reactor, and this will actually then kill the bacteria that is on the equipment. Because normally you would probably heat up the equipment to disinfect it, but that's sometimes not possible with plastic devices, and for this reason you use radiation therapy. So this works for most cases, but the question is, are there some bacteria that are able to withstand that? Yes, even that is possible. That some bacteria are actually have been found in areas of high radiation. So concerning extreme environments, one also has to draw there's a line, of course, a certain limit. It is not possible for bacteria to live in places that essentially would physically or chemically destroy the bonds of the chemicals that they're made of. So, for example, in fire, it's not going to be possible that you will find bacteria, or in lava, volcanoes, not possible. It's too hot. And then also, there's no water present, obviously, because of the heat. Life depends on water. And also the heat is so high that the chemical bonds of the cells would actually break. And this is, of course, then not possible. Now, other microorganisms that can live without oxygen, and that is the next question, and the answer is yes, of course, even that is possible. There are many bacteria that can do that, but even some eukaryotes, some single-celled eukaryotes, protists, are also able to do that. That's a little bit, I would say, probably less common. And bacteria, you don't even have to look very far. In our digestive system, for example, it is a place where there is not a lot of oxygen present. There are a lot of bacteria growing in there, and they're using up all of the oxygen. So it's a very oxygen-poor environment, and there are also so-called anaerobic bacteria living there, who have basically a biochemistry and a metabolism, rather, which allows them to also live in an oxygen-poor environment. Some anaerobic bacteria are strictly anaerobic, and this means that if they're exposed to oxygen, then they're actually going to die. So oxygen is actually, strictly speaking, a very aggressive gas, and if very reactive, and if these strictly anaerobic bacteria in contact with oxygen, then they have a problem, right? So you are able to find these anaerobic bacteria also, for example, in decomposing sludge. Also, for example, sometimes in a pond. If you go deeper down into the ground, that's an anaerobic environment, and sometimes you're able to see from a pond rising some gas bubbles, sometimes methane gas or carbon dioxide gas, hydrogen gas, and these are the waste gases that those anaerobic bacteria, which can be found in the pond, which they produce. And these gases are the waste gases, and they basically bubble out of the pond. So yes, this is indeed possible that there are microorganisms that are able to live without oxygen, and they're not even that few. The next question, how might microorganisms help in the Mars project? So I suppose with a project of inhabiting Mars, and I found 3 possible ways how microorganisms can be potentially useful for actually starting a Mars colony. First thing is we humans need oxygen there, but where do we get the oxygen from? Well, there are so-called cyanobacteria. These are also known as blue-green algae, and they do photosynthesis, and they are able to produce oxygen out of sunlight and water. As a matter of fact, a huge portion of Earth's oxygen is actually made by those cyanobacteria. So this is actually not a new concept. We have it here right on Earth as well. I don't really know where you would put out those cyanobacteria on Mars. Mars is freezing cold. There is no liquid water there. So I don't know how that would be done, right? But yeah, that would be one possibility is to use those organisms to actually produce oxygen. Now, the second possibility would be for the removal of waste products. It's, of course, we humans also produce a lot of waste. And this has to be broken down. And this actually means that the microorganisms will take the waste and convert it to carbon dioxide gas. But this process, of course, needs oxygen, unless, again, you're using anaerobic bacteria to do that. But the removal of waste by decomposition is also very important, and that would be a second possibility. And the third possibility is the use of microorganisms in food production. So, for example, in the fermentation processes, when you make yogurt, or, for example, also for certain other milk products like cheeses or bread, for example, requires yeast. So there are many possibilities how microorganisms have already been used in the past and are still used today in actually making food, and that would be another possibility here. Yeah, so to summarize, on Mars we would need them to make oxygen, to remove wastes, and to make food. Maybe there are a couple of other examples as well. So, and the last question that I was asked here, but then I'm going to go on with some other questions as well. But the last question here is the following: how do tardigrades live in such harsh environments? Tardigrades are also known as water bears or moss piglets. And they are microscopic animals with 8 legs. They're pretty much everyone's favorite because they look kind of cute under the microscope. A little bit clumsy, but they kind of walk around. Actually, look what I got here. I've got here, if you're if you're watching this on YouTube, then you're able to see this is a 3D printed tardigrade. Yeah, in the audio podcast, you're not able to see that. Yeah, but those tardigrades are quite cute to look at. Actually, they can be a little bit aggressive as well. Sometimes some of them are even cannibalistic. They eat up each other as well. But they feed on bacteria and smaller organisms as well, and they are quite remarkable because they're very resilient. So this means they're able to survive dryness. So they're also able to survive heat and cold, and the reason is because they're able to go into a so-called a ton state or into a cyst. So basically, they will slow down their metabolism as they dry up, and they will be able to survive in this inactive form for quite a long time. And then when the conditions are better again and right again, they will basically come back to life. So they are able to do that because in the natural environment where they live, for example, in moss, it's not always moist. There are dry areas and dry times as well. And when the sun is shining and the moss starts to dry out, then they also need to survive somehow. And they have developed this technique of going into this dormant state, and where they're able to survive for a fairly long time. Yeah, I think I read somewhere that they even have sent some of those tardigrades into space to see how they react to cosmic radiation, and they were even able to survive that. So you see that those tardigrades are not only interesting to look at under the microscope, but they also have a couple of very interesting characteristics as well. Yeah, so these were now several questions, but I would like to talk about now another question that I received. And this was a question about a so-called inverted microscopes. So we're moving away now from the microorganisms towards a little bit the technology and the hardware part. And one of the questions that was asked is do you have an inverted microscope, and can you tell us why they are so different? So first, easy answer, I do not have an inverted microscope. And inverted microscopes, they have their optics, the objectives on the bottom and not on the top. And why is that? Because in many cases, those inverted microscopes are used in so-called cell culture observations. It means that you're growing your cells in a petri dish or in a plastic dish, and then you want to see how the cells are growing, and you do not put the specimen on a microscope slide, but rather you put the whole dish directly on the microscope, and you're observing the cells basically through the bottom of the petri dish. So in some cases, there are also special objectives used that have a larger working distance because there is not a thin cover glass on top, but rather a more thicker plexiglass or glass of the Petri dish, which is thicker. So the distance between the cells that you want to look at and the objective has to be larger, and so this also has to be compensated. Yeah, so those inverted microscopes are essentially special purpose devices that you can use to observe the environment. But strictly speaking, they do have I would say probably a more specialized applications in cell culture observations, for example. Okay. Yeah, so it was basically that's basically all I wanted to share with you today. And I simply would like to invite you to also to ask questions and post some questions into the YouTube comments. I will try to pick up on them and I will try to answer them. And today I would like to say again very much thank you that for those questions that were sent to me from a very young, enthusiastic biologist. Okay, and you can see that some of the questions from children can be also quite advanced. Yeah, so I'm very happy that the microscopy and biology is able to interest you so much, and I hope that was able to answer your questions also in sufficient detail. Yeah, and this not only goes to the person who sent me the question, but of course to all of you. I'm going to call it quits again for this episode. I hope that you like this new format. Of course, I'm also still continue to make my live stream every once a week. I've got the regular long form videos as well. So I hope that there is something in it for everyone. But for me, from my side, that's all I would like to say today. I wish you all the best. Happy microbe hunting as always, and see you around again. Bye bye.